Welcome, everybody. In this lecture, we're going to be talking about a lot of the background concepts that dictate host and material interactions. So it's really an introduction to biocompatibility and a lot of the factors that dictate if a material is compatible or not. We're going to be talking about protein adhesion mechanisms, how the concept of different proteins dictate how cells interact with a particular material and how they differentiate into either desirable or undesirable cell interactions. And then we're going to see how a lot of these concepts pop up in later lectures when we start talking about, for instance, blood compatibility of materials or how we evaluate materials in consideration for regulatory approval. We're also going to talk about how host cells react differently to materials under different mechanical stresses. Because all biomaterials, even ones that aren't subject to load-bearing stresses, like, for instance, a hip implant, are still subject to some amount of mechanical forces in the body. And the host responds differently in different cases to that. And then finally, we're going to touch on some of the background concepts that are necessary for understanding tissue engineering, like stem cells. First, I'd like to touch on the differences and similarities between the implantation or introduction of living donor tissue versus non-biologic materials like biomaterials. Because the question we want to answer here is, how does the body respond differently to these two? The first point I'd like to touch on is that the immune system does not respond to non-biological materials. And I say sort of right here because, of course, you can have materials that are incompatible and can cause just horrible reactions to the body. But from the immune system perspective, we have cells that, through evolution, are trained to recognize molecular patterns that are associated with bacteria, viruses. We also have sophisticated B and T lymphocytes that can be trained to recognize very specific antigens associated with pathogens. And we can develop vaccines so that our body learns to respond to these in an appropriate manner. But our body has no evolutionary background for recognizing things like silicones or polyurethanes. So what I really wish to say here is that there are no direct mechanisms for mediating an immune response against non-biological materials, as there are biological or donor tissue living materials. So as we're going to learn throughout this chapter and in future chapters, all host interactions are really mediated by indirect mechanisms. Two of the more common indirect mechanisms could be problems related to clotting. Okay, our body has great ways of getting rid of clots, and we have great cell surfaces that are great for interacting with blood. However, if you introduce a foreign material that doesn't have the right surface chemistry and it doesn't have any anti-clotting factors, then all of a sudden you're going to have problems related to clotting. The second is what's known as the foreign body reaction or foreign body response, which we're going to talk about in detail in a little bit. But really the basis for all these interactions and reactions are the presence and absorption of proteins to the surface of materials. So we're going to dedicate the next half of the lecture to talking about protein absorption kinetics and how they mediate different cellular interactions, because your body has around 700 different proteins in the blood. Some are rather benign and non-bioreactive, like albumin, and some are involved in a lot of biological activities like fibronectin or fibrinogen, and they really can dictate the response of the materials with the host. Now, going back to this concept that implanted materials are immunogenic, I think of materials as being invisible to the body because your body doesn't really care about the foreign materials. They can't see them, they don't react with them directly, and it isn't until proteins come to and adhere to the surface of materials that then all of a sudden your body sees those adhered proteins. And especially if the proteins are adhered in different ways than they would normally adhere to other cells, that's all of a sudden what clicks with the body is saying, okay, hey, this is foreign, right? Normally these proteins aren't here in this concentration, so I'm going to react in a different way than I would normally with my own host cells. Because proteins can act as ligands and integrins, which are adhesion receptors, to interact with cells with. So depending on what proteins are adhered to a surface, different cells are going to then adhere to those proteins, not directly to the actual material itself. And so then understanding what proteins have adhered to that surface and how your material is going to react with different proteins is really what dictates how it's going to react with the host tissue itself. So that's why we do a lot of in vitro and in vivo protein absorption studies as part of evaluating the general reactivity of biomaterials and understanding their biocompatibility. Now, when studying protein absorption, scientists and engineers usually conduct two types of tests, one being adsorption studies and the other being depletion studies. Now, in both types of tests, what is directly being measured is the percent of cells that adhere to a particular biomaterial through indirectly modifying either the material itself and the proteins that are present in a particular solution. In adsorption studies, we're usually looking at one particular protein at a time within a buffer. So it's the simplest of kind of studies because there's only one type of protein present in that particular buffer. And usually what scientists have already outlined at that part is what material they're going to be testing and how they're going to be modifying it. They might do a pre-absorption step where they allow a particular adhesive protein to already adhere to the biomaterial before starting the test. And they also might introduce blocking proteins to see how they affect the adhesion of cells to that particular material. So for instance, on the graph on the left, we're looking at the results of a particular study that analyzed the results of fibroblast-like cells, these 3T3 cells, as they adhere to polymers and copolymers of ethyl methacrylate with certain percentages of hydroxyethyl methacrylate within that copolymer. The three different test conditions under variable amounts of HEMA in this polymer are the cells by themselves in just plain buffer, cells with the addition of adhesion-friendly fibrinogen, and then there's also the introduction of a blocking antibody that prevents the adsorption of any adhesive proteins. As we can see from the results of the graph, without the addition of any adhesive proteins, the cells might adhere to just the material itself in the more hydrophobic-rich EMA polymers, but as HEMA gets introduced, adhesion starts dropping off. However, when introducing the fibrinogen, cells like to adhere to that even in the more hydrophilic conditions of the biomaterial. Whenever we add the blocking buffer, then there's almost no adhesion whatsoever. A second kind of study is called a depletion study. Now in this case, we take a complex mixture of various adhesive proteins, like for instance with using plasma, and then you test the adherence of various kinds of cells in that complex mixture, and then you compare it to that same mixture where one or more proteins have been systematically removed, so in this case depleted, in order to see how valuable or how influential that particular protein was in the adherence of those kind of cells. In this case, by mimicking a complex mixture of proteins like that would be found in plasma, we can test cell adhesion in more realistic conditions like that would be found in the actual body. To understand the benefit and differences between these two types of studies, I want to walk us through a series of hypothetical scenarios and their outcomes on the adhesion of a particular type of endothelial cell. So imagine we test adhesion of these endothelial cells in just a straight buffer, and then that buffer plus fibronectin. In this case, we get an outcome that the cells adhere in the presence of fibronectin. So we could conclude from that maybe that, okay, fibronectin is important for adhesion. Then we test a depletion study, in this case just as a control using the full serum before we've actually depleted anything. And again, we get adhesion of the cells, and we think, yeah, that makes sense, because we saw adhesion with fibronectin. We know that fibronectin is in full serum, so yeah, that makes sense. However, as a follow-up study, you do that same depletion study. In this case, you've now removed fibronectin from that complex series of adhesive proteins, and you still get adhesion. And you kind of wonder, well, wait a minute, I got adhesion with fibronectin in my adsorption studies, yet I took it out here and I still got adhesion, so maybe it's not that important? Sure enough, I follow that up with another depletion study where I remove vitronectin. In this case, I get no adhesion with these endothelial cells. So now I know that within this complex series of adhesive proteins, even ones containing fibronectin, I all of a sudden don't get any adhesion with just the simple removal of one type of adhesive protein. And from that, I can conclude that, okay, in this particular scenario, vitronectin is the adhesive protein that's responsible for adhesion of these endothelial cells. However, if I repeat that same process, only this time I pre-treat my biomaterial with ammonia, which changes the surface chemistry, then all of a sudden the results are reversed, where fibronectin all of a sudden plays a key role in adhesion, and vitronectin, even if we remove it from that serum, appears to have no adhesive factor whatsoever. In another hypothetical scenario, I'm testing platelet adhesion for a particular biomaterial that's going to be used in blood material interactions, and I do separate adsorption studies that tests fibronectin, vitronectin, and von Willebrand factor alone. And in all three cases, I get good adhesion. It's tough to tell what the difference is and which one is more important than the other without comparing them the two. So all of a sudden, I do a depletion study where I only move fibrinogen, and despite the fact that fibronectin, von Willebrand factor, and vitronectin are all in there, I all of a sudden get no adhesion. So you can mislead yourself potentially when only doing adsorption studies if you don't follow up with depletion studies. Because in this case, it turns out that fibrinogen alone seems to be responsible for platelet adhesion, despite the results that I got from my adsorption studies. So there are cases where we want to study just specific protein interactions with cells alone, and there's also cases where we want to look at complex environments like ones that we would see within the body. And remember, some of these hypothetical scenarios that I outlined are only in static cases, right? If we have higher shear rates, like we might see in arteries, we're going to potentially see completely different results. So for instance, at higher shear rates, you can still remove fibrinogen and you're going to get adhesion because van Willebrand factor becomes a much more dominant adhesive protein at higher shear rates. It just has an ability to form and break adhesive bonds with cells much more quickly than fibrinogen. There are also possibilities for other kind of tests where you introduce antibodies that are specific to certain adhesion receptors that effectively block it. And then by blocking those, you can sort of do a pseudo depletion study without actually having to remove that particular protein from the serum. Now, the previous studies I just outlined look at protein absorption indirectly by looking at the actual adhesion of cells that we know bind to those particular proteins. We can also look at the protein absorption kinetics directly and model it. The way these studies are carried out is that typically we introduce different concentrations of a particular protein, and that's represented on the x-axis as concentration in the free media that we subject to a particular biomaterial, and then we measure directly how much of that protein got adhered to the surface, which is represented on the y-axis. The shape of all these adsorption studies results in this sort of sharp spike up initially, and then as the surface gets saturated, higher and higher concentrations have a diminishing effect, and we get this plateau. This shape is modeled using what's called the Langmuir isotherm model, in which theta, which is the percent of your actual adsorption over the maximum possible plateau level, is equal to the concentration of the protein in the free media divided by the sum of what's called the dissociation constant plus that concentration of protein in the free media. Now, to explain exactly what the dissociation constant is first we want to imagine the sort of reaction that occurs between the protein and available binding site S, which results in a combination of that binding site with that protein that's bound to it. Now, in this case, the dissociation constant is like an equilibrium constant. That's the ratio of first the two independent concentrations, so a free bound site and then a free protein, over the concentration of the bound sites. Now, because the bound complex is in the denominator, a lower value for a dissociation constant is going to mean we're going to have more adhesion for that particular protein. So the lower the number, the greater the binding strength is. In this case, we define these binding sites by the amount of mass of proteins that can adsorb to a surface, and usually it's on the order of 0.1 to 0.5 micrograms per squared centimeter of your biomaterial. Now, just like with the adsorption and depletion studies, there's cases where we want to look at adhesion of just single proteins, like how the example I just went over. And there's also competitive assays where we want to compare the relative adsorption of different proteins in combination with each other. Now, in a typical competition assay, you would combine two different proteins at different concentrations, which here, like in the example, is given as a ratio of an unlabeled protein to the labeled protein. And the labeling is just one way of actually seeing the presence of that particular protein. You could have a fluorescent label, a radioisotope label, just some way to actually measure that protein of choice. And then you measure the amount of that adsorbed protein on the surface as a percent of the maximum binding that it can possibly do. So in cases where you have no competition, where there is no B, for instance, you would have the maximum percentage of A, and then as you slowly introduce different concentrations of that B protein, then the amount of the adsorbed A protein is eventually going to drop off as the B protein outcompetes it, until the point where you get a minimum concentration of that A at the surface. Now, the number you want to isolate here is the point where the percentage of your labeled protein drops to 50%. And then you look at the ratio of the two proteins at the point that gives the 50% absorption, and that can give you a sense of the relative binding strengths of both of those in comparison to each other. So in this particular example that I have here on the slide, we're comparing the adhesion of fibrinogen, which is protein A, with an unknown protein B to a particular biomaterial surface. So in case A, the point where they have a 50% ratio of each other is the case where the fibrinogen concentration is 10 times that of B. So despite the fact that B is outnumbered 10 to 1, this still results in 50-50 mixture at the surface. So from that, we conclude that B has a higher affinity compared to fibrinogen. In case 2, where the 50-50 mixture is obtained when fibrinogen and B are at the same concentration, we can thus conclude that they have relatively the same affinity. And then if we look at case 3, where the 50-50% mixture results in the case where the B concentration is 10 times that of A, we can see that B must have a lower affinity because it requires so much more of that particular protein in order to outcompete for the surfaces. Now, in this hypothetical result that I just outlined here, we might have been changing this particular unlabeled protein and then just comparing it to the unchanging fibrinogen. You can also do the same type of test with the same two proteins, but different surfaces, because the different surfaces are going to react with different proteins differently. So now that we're talking about cell adhesion studies and we're going to talk about some of the complex ways that we actually look and analyze cells, let's first talk a little bit about in vitro cell culture and some of the terminology that are associated with that. Now, when culturing media, you need to do in some sort of substrate that can support life of those particular cells. If all you wanted was just bare bones buffer that has some sort of saline component, cells can withstand that for a limited amount of time, but eventually they starve and they need to get food in order to consist and reproduce and live on for whatever studies you're trying to do. So your culture media needs to have many different components that are themselves found in blood or plasma that can help support those cells. So let's go over some of these and what their actual purpose is. So a typical cell culture media is going to have glucose, which is the primary energy source of cells. There's going to be essential amino acids, so ones that can't be produced by the body itself and usually needs to get obtained from a diet. There's also various proteins and hormones that the body themselves would produce, but in just a cell culture media, you won't have these essentially producing functions. And so some of these proteins are just needed for various cell functions. You have a buffering component, which is really just a way of maintaining the pH balance like you normally would in the body. A lot of cell culture media has antibiotics, which the purpose of which is for anti-contamination. All this tends to be frowned upon these days because usually these chemical methods of maintaining sterility or warding off contamination are really just ways of masking improper aseptic technique. So if you have good sterile conditions, you're not going to need antibiotics, and anything otherwise just promotes bad behavior. Outside of the actual media, you need an influx of air, which is important for gas exchange. So your cells are going to produce byproducts. They're going to need to take in oxygen as they deplete it in the actual source. And so you can't just have a rigid and gas-sealed container. You need to have an open container that can facilitate gas exchange. That outside air usually also needs to be supplemented with carbon dioxide, which is really just there to maintain proper pH by the slight absorption of carbon dioxide, which turns into carbonic acid, and slightly acidifying the components of your buffer. To indicate if you're at the proper pH, there's usually also a coloring agent like phenol red added that can turn a nasty yellow color whenever it's too acidic and you've neglected your cells. So visually, you can always see that if you need to turn over your media culture and replace it with fresh substrate. Now, when using cells for studies, cells can be divided into two different categories. One would be primary cells, so these are ones that you've actually gotten from living tissue, and you have to use them relatively fast because they die very easily. Another type of cell source are called generically cell lines, which are immortalized cell lines that have escaped what's known as the Hayflick limit, which is really just a limit of how many times a cell is willing to copy itself before it initiates these programmed cell suicide apoptotic patterns. A very great example of these immortalized cell lines are cancer cells. So cancer cells, really bad in the body, really great in the lab because you can produce them as many times as you want. You can throw them out. You can give them to your friends and your colleagues because you can always just keep making copies of them and the cells are just going to go along with it. They just love staying alive. As opposed to primary cells. Primary cells, it's always tough to get a handle on them. You have to coordinate with someone who's somehow extracting living tissue from a host. So to get human cells, you have to work closely with medical schools and other research labs that have a primary point contact. And then as soon as they get them, it's almost like a race to get those cooled cells over the lab and quickly work with them before they die. Now, when growing cells on a biomaterial, like a petri dish, for instance, in the lab, one aspect you always want to be well aware of is what level of confluency your cells are. Now, confluency is really just the level at which the cells have occupied a particular surface. So 100% confluency would mean that the cells are now occupying the entirety surface of your petri dish. 50% would mean that they're roughly covering half of the area. Now, this is particularly important because as cells start bumping up against one another, they start to undergo what's called contact inhibition, where as they recognize other cells adjacent to them or neighboring them, they start undergoing these processes that inhibit further growth or further reproduction by them. Okay, so if they sense that there's no more space available to them, they're going to stop growing. So if you're trying to make many cells or keep them within a particular growth cycle or growth phase, you don't want to get into this inhibitory phase. And at that point, once they start running out of room, you want to passage them, which is the subculturing of those cells by removing their adherents from that particular petri dish, for example, taking however many you want to continue growing in the next petri dish, discarding whatever you just removed, and then you continue growing them and you maintain them in that particular growth phase. Now, the way you release them from that particular growth media or from that substrate like the petri dish is that you make them undergo what's called enzymatic dissociation, where you use a typical enzyme like trypsin or EDTA, that's when in your passaging solution, and that kind of cleaves the cells from their adhered proteins. Then you can get them free and you can just take aliquots of those cells in suspension and add them to your next petri dish. So it'd be very typical for an engineer to say, oh my God, my cells are at 80% confluency. I better trypsinize them so I can passage them into my next cell flask. Now, so far we've talked about the cells that adhere, we've talked about the proteins that adhere to the surface, and we know that these cells actually attach to these proteins, not necessarily the surface itself. So now let's talk about that attachment process a little bit more in detail. So what cells use to adhere to surfaces or to other cells or other components of the body is that they use integrins, which are adhesive receptors on the cells that bind to particular ligands, like the proteins that we've been talking about. And they're going to be present on whatever materials that you're culturing or your biomaterial. Now, integrins can be incredibly complex, so don't just think of them as anchoring points for a cell to a particular protein, because they're often associated with certain cell proliferation or signaling processes within the cell. So they dictate a lot of the behavior of a cell. So now, because we associate favorable cell adhesion to favorable protein adsorption, there are very common techniques to enhance and promote the binding of certain adhesion proteins to a particular surface. So common treatment methods would be to impart charge to a particular material. One might want to direct adhesion of specific adhesion molecules. There also might be ways to tune the adhesion of certain proteins to surfaces by actually altering surface molecules somehow externally, like either adding functional groups that respond to thermal changes, chemical changes, or maybe electrical or electromagnetic that can both be used to promote adhesion or also detach cells. So, for example, polystyrene is an incredibly common biomaterial used in labware that's associated with cells, but by itself, it's really a poor cell adhesion substrate. However, vendors can somehow pre-treat it either with chemical treatment, like with sulfuric acid, or use ionized radiation that usually results in a charge buildup on polystyrene. And then it makes it a really attractive cell adhesion substrate because so many different proteins want to absorb to it, and that allows the cells to then adhere to it. I've even seen some cool cell culturing techniques that make use of a polymer called PENIPalm, which is temperature dependent. And because of that, under certain conditions at a particular temperature, cells will adhere to it. And then instead of using some sort of trypsinization or ETA chemical to disassociate themselves from the substrate, you can just lower or raise the temperature past the transition point, and all of a sudden it makes the conditions unfavorable for absorption. And then you have a nice monolayer to work with. Now, there are a lot of limitations to biomaterials in the in vitro spectrum. The most obvious of which is the reduced complexity in the in vitro environment, right? We don't have the same in vivo conditions that we would, and we're just trying to replicate as best and sometimes eliminate many of the variables that we might actually should include in order to accurately test our device. A big result of the simplification in an in vitro environment is the lack of any immune or inflammatory response that the body would otherwise create. There are also no real great ways to measure some of these sort of cascading effects that we see, like the vascularization or coagulation processes that are really results of these complex, sequential, and cascading pathways. Now, in the introductory lecture, we talked about the need for characterizing not just the bulk properties of materials, but also the surface properties. Now, the surface chemistry is perhaps the most obvious and in some cases the most accessible parameter control when modifying in vitro conditions to assess the effects on cell behavior. It's also an incredibly complex and dynamic parameter because it affects so many things about cells, like the rate of cell proliferation, cell adhesion, the alignment and polarization of cells, the differentiation of cells, and also the production of extracellular matrices. Now, one of the previously discussed advantages of titanium is that titanium has this great osseointegration feature where cells will actually grow into the material and fuse with it, creating this really hardened interface between the two separate metal and host phases. Now, this can be promoted by blasting the titanium surface with aluminum oxide particles to really roughen it up because the uneven topography promotes the ingrowth of the osteoblasts for bone development. Subsequent acid treatments can also impose a finer and secondary nano-sized sort of roughened terrain that can even further promote this. This is commonly done when a finer amount of osseointegration is required, like that seen in dental implants. Beyond just the surface characterizations, even the bulk characterization of a material can promote different aspects of a cell. So for instance, the elastic modulus of material has been shown to affect the amount of cell adhesion, the spreading of cells across the surface, the migration of cells, the proliferation, and differentiation of cells into more finer and specialized cell lines. So for example, the testing of a particular biomaterial in the presence of neural stem cells can be altered so that with a young elastic modulus can promote the differentiation of these stem cells into neurons, whereas stiffer young moduli can promote these cells into glial cells. And again, the only difference between these two cases was just the elastic modulus. Otherwise, all material properties, both at the surface and bulk properties, were exactly the same. Now, beyond just the mechanical properties of material, we can also look at the mechanical effects and how they can change and alter cell properties. So for instance, mechanical stresses can force cells to align themselves in the direction of an applied load. As you can imagine, this is particularly important for cells in load-bearing applications, like those in the musculoskeletal systems. Now, I'm going to give you a little primer on cell injury and wound repair processes because inevitably when introducing biomaterial to a host, some amount of injury is going to be involved. Whether it's really minor, like you're just introducing a catheter into a vein, or something major, like you're doing back surgery to introduce a spinal fusion plate across two vertebrae, some amount of injury and wound healing is involved in all these processes. So first, let's talk about injury on a cellular scale. And there's two types of injuries that can be introduced in cells. The first is called necrosis, which I like to describe as cell homicide, where a cell is being killed by foreign processes. The two main ways that this can be induced is either by the inability to reverse mitochondrial dysfunction, so a lack of oxidative phosphorylation or a lack of ATP generation somehow. There could also be just a profound and blunt disturbances in the membrane functions of that particular cell. This usually results in a swelling of that cell, and then the body responds to this through rapid mobilization of inflammatory cells to help clean up the debris. So think of this as something has gone terribly wrong and the body needs to respond to it in a really high magnitude. In contrast to cell homicide, there's also cell suicide through a process known as apoptosis, which is usually characterized by a shrinking of the cell. So think shrinking of the cell, apoptosis, swelling, necrosis. Now the big difference here is that apoptosis, because it's actually very common, this is the way that cells kill off themselves if they copy themselves too much, like when we reach the Hayflick limit, there's no inflammatory response. Okay, think of this as happening routinely, all the time, and phagocytes, which are a type of cells that are programmed to engulf other cells, they simply digest the cells and they get rid of them that way. Okay, so this is a completely normal process. Necrosis is not. Now, before we talk about injury responses to tissues, I just want to briefly go over the 4 different main categories of tissue and then give some examples of them. So one type of tissue could be considered epithelial tissues, where we have examples like the skin epidermis, gut mucosal, the retinal epithelium. You could also have connective tissue, like those found in tendons, adipose tissue, bone marrow, and cartilage. There's also muscle tissue, like those found in the hearts or just various limb musculatures. And then finally, we have nerve tissues like that found in the brain. Now, the most common result of the introduction of a particular non-immunogenic biomaterial to any parts of these tissues is what's known as the foreign body interaction or the foreign body response. And really, this is the body's way of trying to isolate that foreign body by surrounding it with a layer of cells to keep it separate from what it considers healthy tissue. Now, in addition to this non-inflammatory response, you could also certainly have an inflammatory reaction to a particular material. And this usually occurs in two different phases. One is acute inflammation, which is really just the initial accumulation of fluid and plasma proteins to a surface, and it's characterized by the abundance of a top immune cell known as neutrophils. There's also chronic inflammation, which occurs later on the timeline, and is particularly characterized by the localization of lymphocytes and primarily macrophages. So in response of massive injury and necrosis of cells, it's these macrophages that are needed to phagocytose. Both neutrophils and macrophages are type of phagocytes. They need to ingest these dead and necrosed tissues and cells in order to clean up the inflamed area. Now, a special type of chronic inflammation results in the formation of foreign body granulomas, which are the result of activated macrophages and other multinucleated giant cells organized around a particular irritating focus. Afterwards, during normal healing and wound repair, there's the generic scarring formation, which is the result of formation of new blood vessels through angiogenesis, deposition of collagen, and the maturation and remodeling of the scar itself. Now, in later chapters, we're going to be talking about blood-material interactions, so I want to particularly give a quick primer on the mechanical forces on blood vessels and their effects on cell differentiation and cell growth and adhesion. There's many ways that we can dictate cell behavior by altering the mechanical forces through flow, or in cases where we can alter it, certainly the maintenance of a particular cell phenotype is going to be influenced by the local and the host mechanical forces that are subject to that particular cell. So all these points bring up the idea of mechanotransduction, which is really just the transduction of mechanical forces applied to a cell or tissue into chemical and biological responses. So there are many different sensors of mechanical forces on a particular cell, such as ion channels, G-protein coupled receptors, intracellular junctions, and then focal adhesion points. And it's also easy to understand, I think, that cells of either the cardiovascular or musculoskeletal system are particularly adapted to these particular sensors and also very sensitive to the particular forces that are subjected to them. So for instance, when engineering cell scaffolds for tissue engineering applications, we know that shear strain can alter the phenotype of cells within those scaffolds and differentiate embryonic stem cells that we put in there into something like smooth muscle cells, if that's what we want. Now, endothelial cells in blood vessels have been studied extensively because they've shown that they have a great ability to respond to changes in shear stress. For instance, under normal conditions, like when you have pulsating, non-reversing shear stresses, they tend to produce more anti-inflammatory and anti-proliferative phenotypes compared to static cultures, right? Because if you imagine a static culture in a blood vessel, seems to indicate that something has gone horribly wrong, right? If blood is no longer flowing, that's going to signal inflammatory responses. That's going to signal the need for cells to grow and perhaps heal. But if you have normal conditions where you have pulsating and non-reversing flow, then yeah, it's going to be no inflammatory responses, no need to grow more cells, everything's going well. There's also been studies that show or compare the difference between reversing, like oscillatory flow, versus non-reversing flow in response to different cell parameters. So for instance, cell alignment isn't going to change whenever there's oscillating flow, but if there's steady flow, cells are going to align themselves parallel to the direction of flow. In sites of inflammation, that's going to be increased whenever you have oscillating flow as opposed to steady flow. And then both proliferation and monocyte adhesion is going to decrease in case of steady flow versus oscillating flow. In part because under oscillating conditions, you're always having changing conditions with the cells. The cells don't know what to do. They're always kind of in the state of these different stimuli that they can't get used to. And so they're always trying to change themselves in that way. They're always trying to proliferate. They're always trying to adhere. They're always trying to induce these inflammatory reactions because they don't know what to do. But under steady conditions, then there's less stimuli to respond to, and that's why all these things are decreased. Lastly, we're going to talk on stem cells. This is going to become more important as we talk about tissue engineering applications. So let's get into it. So the two key characteristics of stem cells, defined by their dual capacity, is the ability for self-renewal and the ability to differentiate. So self-renewal is the ability of the stem cells to maintain their phenotype and continuously divide and reproduce without any negative consequences. Like when we talked about the Hayflick limit and cells undergoing apoptosis because they've been divided too much. Stem cells to certain degrees don't have this, so they can renew as often as they want. And at different stages of stem cells, some can do this indefinitely, and some can just simply do it a lot more than fully differentiated cell lines. The other concept, and I've realized I've said this a number of times already, is their ability to differentiate. So think of stem cells as like a blank slate to become any one of a number of type of cells that carry out specialized functions. And furthermore, there are many different states of differentiation. So think of something like an embryonic stem cell as having the ability to become any one of the 200 plus types of cells in the body. And in order to do so, it goes through many rounds of maturation and differentiation in order to become any one of those specialized cells. So what I'm describing here are different stages of the potency of a particular stem cell. So the ones with the most potency, so called pluripotent, have the capacity to turn into any of the 200 plus cell lines, like I just mentioned. They have an infinite capacity of self-renewal, so they can literally copy themselves as many times as they want. And they also grow and proliferate rapidly. The next step down would be multipotent cells, which have a somewhat diminished capacity for differentiation. So for instance, all blood cells come from hematopoietic stem cells, which are not pluripotent because they can't, for instance, turn into brain cells, but they can turn into any one of the many types of blood cells. So they are considered multipotent. They also have a self-renewal that's at a somewhat diminished capacity, and their proliferation rate is slower than that of pluripotent cells. There are also ones that are further down the line that are unipotent, which are similar to multipotent in that they have good self-renewal capabilities and differentiation abilities, but they can only differentiate into a single cell line before they're terminally differentiated. So I think that's a good summary of the different cell functions we're going to be talking about, the need to evaluate material surfaces and bulk properties and how they affect cell adhesion to these materials, and then also some of the mechanisms that dictate cell adhesion, particularly the type of proteins that it adhere and what can influence protein adhesion and how we actually test for that. So thanks for listening.